Hi everyone, my name is Vishnu Prabhu. I'm an assistant professor here at the University of Central Florida in the School of Modeling, Simulation and Training. Today we are going to present our paper, Psychological and Neurophysiological Indicators of Stress and Relaxation in Immersive Virtual Reality Environments. The work is primarily done by my prior students who are the first four authors in this particular paper. And the work was conducted at prior University of North Carolina at Charlotte. We also have a collaborator from the Cleveland State University, Dr. Apostolos Kalatsis. So moving on, what motivated this particular study is just the evolution of VR that we have seen over the last couple of decades, where VR has gone beyond just the immersive display technology to kind of the gateway for human-computer interaction. In its current era, VR enables us to modulate perceptual, cognitive, and affective domains across the participants. And from just my area of research, which is primarily healthcare, its ability to induce different affective states or modulate different affective states, particularly in its control settings, have made it one of the most exciting things in the last couple of years. And the growing interest of affective VR systems that detect, interpret, and adapt to human emotional state has also been investigated a lot lately. And one of the primary challenge that we are facing right now is for us to develop these affective aware systems, we need to know the neurophysiological underpinnings, like what changes or how human physiology changes when they interact with certain VR environments. And the challenge to kind of understand the neurophysiological changes in the human body when they interact with the VR is we primarily rely on survey methodologies. Validated survey tools such as state rate anxiety or visual analog scale help us to understand the level of stress, pain, anxiety, different things. But they have their own limitations, such as recall bias, limited temporal resolution, because we collect the data pre and post, or at certain time periods, and we are not collecting it from a real time continuous perspective. And that's where we have seen a lot of the development in biosensing come into play, such as we are able to collect EKG or PPG in all the smartwatches or our rings and things like that. But still, there is another particular research gap that we have noticed in literature reviews that most studies look at one particular setting. That is, conduct a study how this particular relaxation environment affects participants or stress-inducing environment affect participants. There is not a single study, to our knowledge, that we have seen where the same set of participants were exposed to different set of environments. And that's what we are trying to do in this particular study. So in this case, we looked at relaxation intervention, passive stress induction, and also active stress engagement. And I'll get into more details how we did that and what we did to induce these three particular emotions. The two specific research questions that we are trying to address in this particular case study was, can we measure and compare neurophysiological responses again across those three settings? That is relaxation through heart rate variability biofeedback, then passive stress through a parkour video, and active stress through a Richie's plank, which is an out-of-the-box tool. So we are trying to see how human physiology changes when they are exposed to these three environments in virtual reality. And the second question that we are interested in was looking at the objective neurophysiological responses and how do they correlate and how do they correlate with the subjective responses that we have. So to kind of quickly give an idea of our multimodal approach in this particular research, so we have two survey metrics, that is state rate anxiety and visual analog scale. The first one collects anxiety, the second one we use for both stress and anxiety. So we deployed VAS twice. And from a physiological measure standpoint, we have three different measures. That is brain activity, that is the prefrontal cortex, so FNR system, then cardiac activity using HRV, and respiration patterns. And you can also see the different devices that we used here. And we also show the image of participant outfitted with all this data, all these measures, and you can see more details in the paper regarding how the sampling rate and all those. But to give a quick idea, this is the FNR system that collects the front part of brain activity. The strap here collects the breathing rate, and also we have the EKG system connected with the participant. And in terms of study design, we had participants engage in all these three different settings. So it was a repeated measure study. And we had a total of 14 participants, so again, a small sample size. And all participants in this study were South Asian demographic. And we ensured the to avoid any biases in terms of ordering. We counterbalanced the order. So in sequence one, a participant could get forest, parkour, and plank. In sequence two, it could be parkour, plank, and forest. And between each of these sessions, we gave them a 6-minute break to avoid the carry forward effect that we often see with the neurophysiological data. So we can keep the data clean. So just to give a quick idea of what the VR environment looks like is you can see here, let me show the picture as well. So this is something we developed within the lab using Unity software, and we connected it to the breathing rate of 6 breaths per minute. So our optimal range is 5.5 to 6.5, and we gave them multiple feedback. So on the left hand side, you see a foggy environment. So that's when a participant is not breathing in that range, and you can see they are currently breathing at a higher than the optimal range. But once they keep it in that green area, the fog would also remove. So they are getting multiple cues along with an audio cue as well, which we currently do not have in the picture, but it would be available later. Then the second intervention was a parkour video, and we call it a passive stress injection environment because a participant does not actively engage in this video. They're watching a parkour video, which is when participants jump from a high building or a high-rise building, and they're just viewing it from a point of view perspective. So that's why we call it passive. And the third one is what we refer to, or not we refer to, it's the Richie's Plank experience, which is an out-of-the-box tool, and it's a validated tool, and it's used for inducing stress. And we call this one an active stress environment because here participant would walk on a plank. In lab, we had a plank where they would walk on, and then when they look down and they're kind of pushed off the plank. So the stress induction here is a bit more realistic, and that's why we refer to this as an active stress environment. I'll quickly move on to results, and I'm going to segment it by subjective and objective data. So with subjective data, what we saw with stress was both Richie's Plank and Parkour video induced stress compared to the pre. So comparing to their pre scores, both of them induced stress, but we did not see any significant change in the forest environment, and we observed the same thing for anxiety. And the primary reason we did not see any change with forest environment was their stress and anxiety levels were already lower prior to the engagement. So there's no further reduction that could happen. So that's why we did not see a significant decrease or a statistically significant decrease. However, what we found interesting from a data standpoint was that across group comparison, where we looked at stress from parkour and also Richie's Plank to the biofeedback forest, we saw that participants in biofeedback forest had a lower levels of anxiety. Moving on to respiratory rate, this is kind of given because we do expect this to happen, where in biofeedback we are asking participants to breathe at a lower pace. And as you can see, what we see here is although the ideal range we are targeting was 5.5 to 6.5, on average it was 11.2 because it's also challenging to breathe at that particular rate, but it significantly varied from all the other scenarios, that is baseline, Parker, and Rich's Plank, which all had a higher breathing rate. And finally, looking at heart rate variability, I'm not going into the depth of what RMSSD and what LFHF ratio. One is time domain metric, the other is frequency domain metric. But what you can see here, or what is interesting here, was that Ritchie's Plank had a significantly higher activity compared to the other scenarios. And also, in general, what the observation we noticed here was Ritchie's Plank induced more. Um, more stress, more anxiety, often measured as fight and flight response. So that's the parasympathetic and sympathetic activity that we see here, and the LFHF ratio values that you see here. And finally, moving on to the brain activity, that is deoxy and oxygenated hemoglobin activity. What you can see here, we first present the oxyhemoglobin activity, and we divide the frontal part of brain into 4 different areas: left lateral, left medial, right lateral. And in all these cases, we saw the highest activity of HBO, that is the blood flow, during Ritchie's Planck experience compared to the forest. And looking at HBR, we only found significant difference in 2 areas. but the results kind of inverts here, where the lowest activity of HBR was seen in Rich's Planck and the highest was in Forest. Overall, what we are observing here is Rich's Planck experience in general induced a lot of cortical engagement, or the blood flow in your brain in that particular areas significantly increased compared to other scenarios that we are looking at. So what are some of our key findings? What did we learn from this, again, proof of concept small study group was that stress conditions, both Parker and Planck, increase subjective anxiety and stress. We also saw elevated respiratory activity and also a high prefrontal HBO activity, meaning more cortical activation. And relaxation, we saw reduced biofeedback effect of reduced respiration rate. We also saw a lowest prefrontal activation, and we can see a different sympathovagal pattern compared to the stress environment, both of the stress environments. And what we are hoping to contribute, or the primary outcome that we are contributing from this research is, one, it's an integrated data set that would be available for public, where we have both subjective and objective measures, that is synchronized across multiple VR environments. Now, this is super useful in future when we want to develop machine learning algorithms that we want to test or to classify stress or anxiety compared to a relaxing environment, and also the neurophysiological profile. So the raw data could help us understand how it changes. Is there any particular prefrontal activity signatures that we can learn from the brain activity that we are collecting here? However, we do acknowledge there are several limitations in this particular study that we conducted, and the primary one is just the small sample size we have. Although we collect the data over a longer time period, 30 minutes for each participant, it's still the variability among participants are lower, and also the homogeneous demographic is one other issue, and we acknowledge this in paper as well. And also there were some movement artifacts during Richie's Planck experience, which we also acknowledge and we are aware of. But that still doesn't mean what we learned from this study cannot be scaled or implemented. So again, the main conclusions that we have is the multimodal approach that we have used. We saw there's significant difference. Going back to our research question, there's a difference in neurophysiology activity as well as the subjective responses when they are engaging in different VR environments. We also saw the values correlating, meaning the subjective responses and objective responses align, and each VR scenario produced a distant or a particular or a distinct neurophysiological signature. This would help, like I said, to develop the future, ML algorithms or other algorithms that we can use to compare or develop new particular scenarios for personalized interventions. However, again, we are cautioning, this data that we see here is from a small study of 14 participants. Hopefully, we can scale this to a larger group and also more diverse group, so we can learn more from this, what the neurophysiological signatures look like and patterns would look like. And if you have any questions, you can reach out to me directly. The email is available on the paper. And I hope you all have a great conference as well.